Another happy day at bed. So like singing. Little tune. Okay. Hi everybody. So we are delighted and honored to have Dr. Bridget Callaghan joining us here. She's an assistant professor in the Department of Psychology here at UCLA. She is also the group lead for mental health disorders at the Goodman-Luskin Microbiome Center, the new fancy microbiome center here at UCLA that we are very lucky and special to have in this cutting edge area of research. She started her lab here in 2019, so we're going to hear a bit about what she's been up to and some of her recent endeavors. So please join me in welcoming Dr. Callaghan.
Thank you. So the microphone, it's not amplifying, right? Like it doesn't need to amplify, that's fine, it's all good. All right, so today I'm going to talk about the intergenerational impacts of adversity on mind-body health and specifically looking at pathways through interoception and the gut-brain axis. So what I'm going to attempt to do is to present almost all new data that has been collected in the past few years since starting here at UCLA. So this is also the first time I'm presenting a lot of this data. So we'll see how we go. All right, so my research program is really centrally focused on this relationship, the attachment relationship that exists between a parent and child. A relationship that, when it is secure and strong, can really help children deal with all manner of challenges that they may encounter in this world. And when it is disrupted or ruptured, can really predispose individuals to exhibit difficulties across basically all areas of their life. For example, adverse childhood experiences, or ACEs, particularly those involving maladaptive family functioning, account for up to 30% of adult mental illnesses. And these same types of experiences are also associated with an increased risk of basically all examined physical health outcomes, including things as diverse as heart disease, respiratory disease, and then also health risk behaviours like risky drug use. So in other words, from this literature, we can see that caregiving adversity really affects the whole child and their development, sticking with them throughout their lifespan and potentially even into the next generation. So we know that there are generational effects of ACEs through several studies showing that seriously traumatic and horrifying events like genocide, the Holocaust, 9/11 survivors impact the mental and physical health of those who are directly exposed to the events and also impact the mental and physical health sometimes of their children. So this shows us two primary things. Firstly, it shows us that continuing to treat mental and physical health separately is only ever going to get us part of the way to the health and well-being that we are all striving for. And it also tells us that understanding health and well-being really needs for us to take into account the experiences that we have had across our lifespan, as well as potentially the experiences of our parents and maybe even our grandparents as well. So my research program really focuses on those two components, so physical and mental health and these generational impacts. So specifically in my research, I ask what are the mechanisms by which caregiving adversity in one generation become biologically and psychologically embedded within an individual, sticking with them throughout their life to affect the brain behavior biology, that is the physical and mental health of the next generation. So in today's talk, I'm going to present some research demonstrating the intergenerational impact of caregiving adversity on the mind and body. And I want to spend just a moment now describing the two types of caregiving adversities that I focus on most heavily in my work. So the first of these is institutional care in either baby orphanages, baby hospitals or orphanages abroad, followed by international adoption, which even under the very best of circumstances or conditions is a very impoverished caregiving environment characterised by a really high child to caregiver ratio and the lack of a stable and consistent caregiver. And the other experience is foster care within the United States, followed by domestic adoption or a guardianship care arrangement. So beyond the circumstances which would lead a child to end up in foster care in the first place, these youth are also experiencing separation from their parents and sometimes their siblings and other family. And it's a profoundly challenging experience for these children from many of those different perspectives. So obviously these are two very, very different types of experiences, but critically for our purposes, and certainly for the purposes of the talk today, they are similar in two important respects. Firstly, they are both really significant caregiving adversities. And secondly, both groups are in a stable caregiving arrangement by the time they participate in research, which allows us to, the best of our ability within a human sample, try to isolate the impacts of these early caregiving environments versus ones that happen later on in development. So I'm going to present data from several studies in humans demonstrating how early life adversity gets under the skin to influence children's emotional and physical health, paying particular attention to the brain and gastrointestinal system as they participate in the brain-gut microbiome axis. And the reason I'm interested in focusing on this system is because of the many, many studies in both rodents, monkeys, and humans showing that this axis is really critical for many aspects of mental and physical health. And then I'm going to show you how these experiences of adversity can affect the next generation via changes to the brain and emotional health of youth, as well as to their microbiomes. And then finally, I'm going to zoom in on pregnancy to examine some mind-body mechanisms that might be involved in the perpetuation of childhood trauma across generations. So without further ado, let's start at the first arm of this model, looking at adversity impacts on emotional health. So for this part of the talk, I'm going to highlight work from a wonderful postbac, Genesis Flores, who's now at USC, and a fourth-year graduate student, Fran Kodasi. So when we think about adversity impacts on an individual's emotional health, we really need to think about this concept of multifinality. That is, that people can end up at different places after having a similar type of caregiving experience. So early life caregiving adversity is characterized by very diverse outcomes. So anxiety, depression, developmental disorders like ADHD, psychosis, all of these have an increased incidence after experiences of caregiving adversity. In fact, this really interesting recent study suggested that heightened stochasticity within the developing brain may serve as an adaptive mechanism under conditions of environmental uncertainty, which is certainly what early caregiving adversity is. So one thing that has really interested us in the lab is to think about components of emotional health that are affected by adversity, but that might help to explain the diversity of symptom profiles that we see after adversity, so-called transdiagnostic mechanisms. And for that reason, we've been very interested in thinking about alexithymia. So alexithymia is comprised of difficulty identifying and describing feelings, difficulty differentiating between emotional states and physiological sensations, and a really concrete, externally oriented style of thinking. And it is a transdiagnostic risk factor for a range of different disorders, like internalizing, externalizing, somatic disorders, as well as developmental disorders. Now, most of the research that has looked at alexithymia has tended to focus on adulthood. It's thought to be some sort of a trait-like mechanism. But developmental theories have emerged that suggest that the attachment relationship is really critical in building up emotional health to kind of veer individuals away from trajectories of alexithymia. And in fact, in adults, there was a recent meta-analysis on over 30,000 adults showing that emotional abuse, emotional neglect, and physical neglect, things that affect the attachment relationship, were associated with adult alexithymia. So in this first part of the study, what Genesis and Fran wanted to do was to examine the association between adversity exposure and alexithymia in a sample of emerging adults. So here in this community sample, we had 355 18 to 25-year-olds. And the reason that they wanted to focus on this age group first was because it's very much neglected in the literature so far, and it is a developmental period in which there's a high emotional vulnerability and lability. So these are, youth who are, or emerging adults who are, the types of people that we see in our undergraduate labs. And I'm sure I don't need to tell any of you that they're really struggling emotionally, particularly after COVID. So this was a really simple study. So Fran and Genesis went ahead and asked these individuals to report on a range of different early life caregiving experiences on 3 different measures. They ran an exploratory factor analysis and found support for a 3-factor model, so sexual abuse, physical abuse, and then this broader factor of family dysfunction. And when they linked these different factor scores to alexithymia, they found that only family dysfunction was positively associated with alexithymia in this age group. So following that up, what Fran and Genesis did was to look in a sample that we had collected in the lab of 157 6 to 16-year-olds who had a verified form of early life caregiving adversity, which very much hits on family dysfunction, foster care versus a comparison group of individuals who had always been with their birth first or biological families. And so what they find across age is that, again, this might be a bit hard to read the text, but in blue is the caregiving adversity, scoring higher on the alexithymia scale. And we also found in this study that greater time spent in unstable care, so an older age entering stable care, was positively associated with alexithymia. Within the early life adversity exposed group, we also found that child-reported feelings of security in the relationship with their adoptive or guardianship caregivers was negatively associated with alexithymia. So really speaking to this idea that forming stable bonds, even in the post-adoption environment, can really help youth set up a good foundation from which to build these basic building blocks of emotional intelligence. On a subset of these individuals, we also had data on how caregivers and children interact while they were discussing a conflict, so a negative event, and we coded caregiver behaviour for supportiveness towards the child during that interaction. And what they found was that while there was no association between caregiver supportiveness and alexithymia when they were measured contemporaneously, they did see that parent supportiveness was negatively associated with alexithymia measured 2 years later when accounting for alexithymia measured at time 1. So this kind of suggests to us, particularly using a behavioural measure of parent supportiveness, which I think is particularly strong in this study, that these parent behaviours may be actively shaping the emergence of emotional behaviour in youth. So to summarise this section, I've shown you that family dysfunction is associated with heightened alexithymia in young adults, that maltreatment followed by foster care, which is a significant caregiving adversity, is associated with elevated alexithymia in children and teens, and that the quantity and the quality of caregiving matters even in the post-adoption environment. So alexithymia was associated with a longer time in foster care, lower relationship security with caregivers, and less supportive parenting in the parent-child interaction. All right, so now that we've looked at that first association with mental health, I wanted to switch gears a little bit in this next part of the talk and focus on physical health. So for this part of the talk, I'm going to highlight the work of Dr. Paul Bloom, who is now a postdoc at Columbia University, and Nomi Gantz, who is a third-year graduate student in the lab. And the physical health complaint that we're going to focus on in this section is gastrointestinal distress. And the reason that we are looking at GI distress in the context of adversity is really because of disorders like irritable bowel syndrome. So irritable bowel syndrome, or IBS, affects 1 in 10 people globally, making it one of the most common disorders of gut-brain interaction across the world. And it holds a significant disease burden for the society, for economy, and also for individuals who are affected with it. So for example, this Drossman study found that adults who had IBS were willing to give up 25% of their remaining lifespan, which was an average of 15 years, to be symptom-free. This is a really significant problem that people deal with. It really affects their lives. Now, not surprisingly, given that statistic, IBS is very comorbid with mental health problems in adulthood, particularly anxiety. And trauma is also present in many of these adult cases of IBS. So up to 80% of adult IBS cases also have a history of trauma, with an emotional abuse being the strongest predictor. Now, critically, like alexithymia, IBS very likely has developmental origins. So for example, adults who recalled having abdominal pain as children were 2.5 times more likely to have IBS as an adult. Moreover, because IBS is a disorder of brain-gut interaction, this may suggest that these types of disordered interactions may emerge early in life, and that this might be a particularly interesting biological system to look at in the context of adversity. Okay, so we've seen in several studies now, using different samples, different questionnaires, parent report, child report, caregiving adversity, be it institutional care, foster care, or other, are associated with heightened gastrointestinal distress. So here I'm showing you just one graph that's taken from our most recent study of 160 youths aged 6 to 16 years old. Here the caregiving adversity is foster care, and you can see that these self-reports of gastrointestinal distress are higher in this population. In Dr. Paul Bloom's work, we've also shown that GI distress goes consistently hand in hand with mental health, even very early in development. So here, in 3 different cohorts, he demonstrates a remarkably similar association between gastrointestinal distress and anxiety in youth. So everyone here are less than 18 years of age. And importantly, in this larger cohort, the Healthy Brain Network of nearly 2,000 individuals, they actually had anxiety that was reported not just by the parent, but also the child in green, and clinician consensus diagnosis of anxiety disorders, which is the purple graph all the way over on the right there. So you can see that this association is also remarkably robust to responder. So one reason why adversity, anxiety, and GI distress may go together, particularly early in development, is because of the well-worn connections between the brain and the gut via the brain-gut microbiome axis. Indeed, we know that bugs that live within the gastrointestinal tract are responsive to cortisol and HPA axis activation, and are known to influence the brain via spinal and vagal afferents, as well as the production of neuromodulating metabolites, amongst many other very interesting mechanisms. So one significant part of the research program in the lab over the last several years has been to try to understand what the impact is of these microbes early in life, and then, so what the impact of adversity is on these microbes early in life, and then in turn to try and understand how microbes affect the brain in ways that contribute to mental and physical health. So let's have a look at how adversity impacts the microbiome in youth. So we've actually already published a study showing the impact of caregiving adversity on the microbiome before. That was in youth who were internationally adopted from institutional care settings, but it was a very small sample size. We did show impacts there, but it was only 16 youth, because at the time we were starting this, it was very new sort of research to do. It was pretty hard to get money to do anything like this, and it was outrageously expensive. It remains outrageously expensive, but now we have some money to do it. So what I'm going to show you are unpublished data that Nomi has in prep on data that I collected this data as a postdoc in New York. So these kids are 2 to 17 years of age with a mean of around 8.5, and they were either domestically adopted from foster care in the US or internationally adopted from institutional care settings. So we've kind of lumped those together. They don't really seem to be too different. And then we've got a comparison group of youth who are always with their birth first or biological families. So when researchers look at the microbiome, these are 3 of the most common measures that researchers use. So the first one is alpha diversity, which is a within individual metric. So this is me asking how rich and evenly distributed are the microbes in my gut, in Molly's gut, in everyone else's gut, and then let's measure some variable like early life adversity and see whether, group people together and look at mean differences in adversity exposed and not. Beta diversity is a between individual metric. So this is where we look at features of my microbiome and I'm going to compare them to Molly's, and to everyone else in the room, everyone else is compared to everyone. And so we're going to get this big matrix of pairwise distances, and we will use something like adversity to explain variance in those pairwise distances. And then finally, differential abundance is where we look at all of the individual microbes at the kind of lowest taxonomic level that we can get to, which is usually the genus level. We do a whole bunch of tests looking at adversity versus not for all of those different microbes, and then obviously we correct for multiple comparisons. So in this study, Nomi found impacts of adversity on each of these different microbiome metrics. So looking firstly at alpha diversity, Nomi found a really interesting interaction between caregiving adversity and age on Faith's phylogenetic diversity. So Faith's phylogenetic diversity is basically just asking how related are the microbes in my gut to one another. So how many branches of the phylogenetic tree do they share? Usually what we see in early development is a massive increase in alpha diversity from 0 to 4 years of age, and then a stabilization of alpha diversity from that point onwards through to like 80 years of age. And that's what we see in the comparison group. So there's no significant change with age within the comparison group in terms of alpha diversity. But what we saw with the adversity exposed group was a significant nonlinear decrease in alpha diversity with age. So this is a really unexpected finding, to see any interaction with age in this age range, so 5 to 16 years old or 6 to 16. In terms of beta diversity, we also found that this was associated, or rather we found that adversity was associated with differences in beta diversity. So here we're plotting each individual on these 2 principal components, and the circles are covering all of the data that are within a particular group, with comparison in red and adversity in blue. The fact that these circles are overlapping suggests that the centroids are the same, but that their different sizes suggest that there's differential dispersion along these 2 principal components as a function of early life adversity. And then finally, in terms of differential abundance, I don't expect you to see this, but just to kind of describe what this is, we looked at adversity main effects with a whole range of covariates. Also important to mention, we had a bunch of covariates here, including diet and breastfeeding and birth mode and things like that. The next plot is an adversity by linear age interaction, and then the final one is an adversity by quadratic age interaction. So I'm just going to zoom in. Whoa. Okay, I'm not going to zoom in. All right, I am. I'm going to zoom in. I'm going to zoom in on those interactions. So anything that was only linear and not quadratic is shown in the top little panel, and everything else is shown in the bottom. And so what is the point of showing you this graph is to show you firstly, the microbiome has definitely not finished developing by around 4 years of age. I keep hearing that over and over again, and that's certainly not true. I mean, even if we take just this top one here in the comparison group, there's pretty significant change across this age range. So that's the first important point here. And then secondly, it seems that adversity is changing these developmental trajectories, and it's changing it in both kind of linear and non-linear ways. So the next thing that Nomi wanted to do here was to see whether any of these held any kind of functional significance in terms of outcomes that we're interested in the context of adversity, like internalizing disorders. So within this set of microbes that were significantly associated with adversity in some way, Nomi then tested, correcting for multiple comparisons, whether any of them were associated with internalizing disorders or symptoms, and she found that these three were. So specifically, she found that Clostridium inocuum, which is typically a pathogen, was positively associated with internalizing disorders, and this is controlling for caregiving adversity group age and their interaction. Eubacterium hallei utilizes glucose acetate and lactate to form butyrate. Usually we think of butyrate as being a generally positive thing. Here it's positively associated with internalizing disorders or symptoms, so that was surprising. And then finally, this butyromonas is a butyrate producer, and as expected, it was negatively associated with internalizing disorders. So some sensical associations here, some kind of confusing ones, and I think that really highlights where the microbiome field is at the moment, particularly within human studies where we're dealing with these types of taxonomic data. So it's really tough for us to get anything lower than the genus level, but there's huge, tremendous diversity within the genus level. There, in one genus you can have bacteria that are associated with butyrate and also not, and that are pro-inflammatory, anti-inflammatory. So I think this really speaks to the idea that we think these microbes are important in some way, and we need to do a lot more work using different sequencing techniques, particularly metagenomic sequencing, to get a good idea of the functional significance of these, and so we're doing that now. Now, I've spoken a lot about the gastrointestinal microbiome, which is definitely like the darling of the microbiome world. Everyone loves the gastrointestinal microbiome, but of course it is not the only microbial community that exists. So Nomi and I have been really interested in thinking about the impacts of adversity on other microbial communities. So microorganisms that inhabit the oral cavity are the second largest community of microbes in the human body after the gut. And because they're in the mouth, that means they're situated at the primary gateway for toxin, nutrient, and pathogen entry into the human body. And because they're connected to the gut through the alimentary canal, they also are associated with the gastrointestinal microbiome. So this is a really important site of influence for health outcomes. Now, these microbes can affect the brain through a variety of pathways, including translocation to the periphery, causing systemic inflammation, translocation to the oral microbiome, causing changes to the oral microbiome, which then impacts the brain. And then also they can directly travel to the brain through the trigeminal nerve. And then there are also pathways of influence from the brain to the oral microbiome. So again, systemic inflammation, gingival inflammation, it changes the structure of the mouth and therefore the microbes that can survive there. Similar types of impacts with gonadal hormones. Critically for early life adversity, cortisol also affects oral microbes, and also health behaviours, which are also linked with early life adversity, impact the oral microbiome as well. So we wanted to have a look at how early life caregiving adversity impacted this community. Now, we weren't the first people to think of this. In fact, there is already a study published in 2021 in adults retrospectively reporting on their experiences with childhood adversity. In this study, they found no influence of alpha and beta diversity, but they did see a range of differentially abundant microbes in the oral cavity as a function of caregiving adversity. So Nomi was really interested in looking at these impacts earlier in development with a verified form of adversity, and she particularly wanted to zoom in on these pathways that intersect with the stress hormone cortisol, as we know that cortisol is dysregulated in the context of early adversity. This is one study, there are many that have shown this effect. And so she had this really interesting hypothesis based on these studies showing that immune cells are actually less sensitive to the regulatory effects of cortisol in the context of caregiving adversity. So Nomi's hypothesis was that potentially the same would be true of the oral microbiome community. That is, that we would see interactions between how the oral microbiome how cortisol influenced the oral microbiome community in the context of early life adversity. So in this study, she had 6 to 16-year-old kids, half of whom were in foster care and the other half, or had been in foster care, and the other half were a comparison group. And she had saliva microbiome and hair cortisol data. So hair cortisol is really reflecting the past couple of months of kind of chronic cortisol exposure. And so again, Nomi found differences in alpha, beta, and differential abundance with a lot of interactions with cortisol. Okay, so firstly, Nomi at the level of alpha diversity found, again with Faith's phylogenetic diversity, the one that we were looking at before, an interaction with caregiving adversity and cortisol, whereby there was a negative association between hair cortisol and Faith's phylogenetic diversity in the comparison group, and as she expected, no association between cortisol and Faith's phylogenetic diversity in the adversity group. Okay, so then in terms of beta diversity, which is the between individual metric, this is using a metric called Jacquard dissimilarity, which is basically the presence or absence of microbes. And here again, she sees that there's an association, or cortisol is explaining variance along these principal components within the comparison group, but not within the caregiving adversity group. So a fairly consistent story across these two metrics. For differential abundance, Nomi didn't see an interaction with cortisol and caregiving adversity, but she did see that caregiving adversity, there was a main effect where it was associated with a feature identified as Rothia mucilinagosa and a feature identified as an uncultured porphyromonas, and both of these were higher in the caregiving adversity exposed group. And critically, the porphyromonas amongst these two was uniquely and positively associated with children's internalizing disorders. We don't know that much beyond this about what really porphyromonas kind of means functionally in the environment. Okay, so the take-home message for this section is that adversity is associated with physical health complaints like elevated gastrointestinal symptoms. We've also seen a range of other health complaints, including somatic symptoms like aches and pains and rashes, that are also elevated in the context of early life adversity. Adversity also impacts the composition of the gastrointestinal and oral microbiome communities, and these microbiome changes might underlie some of the mental health complaints seen after adversity. We haven't looked at their associations with physical health yet, but we think the same may also be true there, but potentially in different ways. Okay, so now that we've looked at the impacts of adversity on physical and mental health, now I want to switch gears a third time and think about how these effects might be perpetuated across generations, so looking at these intergenerational impacts. So for this section, I'm going to highlight the work of a wonderful former postdoc, Dr. Jessica Uy, who is now doing a second postdoc in Ian Gottlieb's lab at Stanford, and my fourth year graduate student, Frank Kadasi. And for both of the studies I'm going to show you here, we actually worked with an amazing collaborative team to use this exceptional data set called Growing Up in Singapore Towards Healthy Outcomes, which is dyadic data, mothers and their children, so a second generation, and it has data across just so many different variables. They're really beautifully phenotyped. So in this first study I'm going to show you, we were looking at the impacts of adversity across generations on brain and behavior. And for this study, Jess had access to 541 dyads. The first measure we had was mothers' experience with childhood trauma, which was retrospectively reported by mums when they were pregnant with the focal child. Maternal prenatal mental health symptoms, so maternal prenatal depression and anxiety can be considered as somewhat of an adverse event for the fetus or an adverse environment for the fetus. Maternal postnatal mental health, so again, depression and anxiety in mothers is generally tended to be thought of as a challenging event for a young child. And then children's emotional health at 7 years of age, which was both parent-reported, proxy-reported, and child self-reported. So Jess found, firstly, that maternal childhood trauma was related to worse parent proxy-reported child emotional health at 7 years of age, and that this occurred serially through a pathway of worsened maternal prenatal and postnatal mental health. She also saw that maternal childhood trauma was related to child self-reports of emotional health, and that this occurred through a pathway of worsened prenatal maternal mental health symptoms. Now, in a much smaller subset, so they didn't scan all 500 and we only ended up with 89. They scanned a smaller subset, and then of those we chose the highest quality resting state data. So we had 89 resting state amygdala-seeded data here. And what Jess found is that over and above maternal childhood trauma and maternal prenatal mental health, maternal postnatal mental health was uniquely associated with children's amygdala, prefrontal cortex, resting state functional connectivity. Specifically, worse maternal mental health was associated with more negative or lower amygdala PFC connectivity. And in turn, she found that this amygdala PFC connectivity pattern was associated with both parent proxy and child self-reports of emotional health when children were 7 years of age. So here, brain scans at 6, and the behavior is taken at 7 years of age. So this suggests to us that there are intergenerational impacts of adversity on emotional health, and these seem to be, at least in part, mediated through the impacts of these early life maternal environments on mum's adjustment to parenthood, so mum's mental health in the prenatal and postnatal period. So in what other ways might generational adversity affect children? Well, we were very lucky in this study, or in this data set, to also have microbiome samples from many of these children. So here I'm going to show you Fran's study, which was recently accepted at PNAS. And so here Fran was looking at a fairly similar model, but replacing the outcome with child microbiome at 2 years of age. So maternal childhood trauma, maternal prenatal mental health, and then Fran actually expanded the postnatal mental health measure, and she used postnatal mental health plus negative life events that had occurred to the child from birth till 2 years of age when the microbiome was sampled. Okay, so what did Fran find in terms of the microbiome? Firstly, in terms of alpha diversity, she didn't see an impact of maternal childhood adversity on child's alpha diversity, but she did see an impact of prenatal and postnatal adversity exposure. However, it was in kind of different and perhaps confusing directions. So firstly, she found with prenatal adversity on the left here, there was greater evenness in the microbiomes of 2-year-old children in the context of higher prenatal adversity exposure. Again, I think this highlights some of the difficulty with doing this work across development, in that while evenness is generally thought to be a positive thing in adults, we really don't know whether it's positive or not in 2-year-old children. So maybe it is, maybe it's not, remains to be seen. And then in terms of postnatal adversity exposure on the right graph, using Faith's phylogenetic diversity, she found lower levels of Faith's in the context of high postnatal adversity exposure. For beta diversity, no differences beta diversity was not explained by any of these adversity exposures. However, each of the adversities was associated with a range of differentially abundant microbes. So I'm going to show you them in this log two-fold change graph here. So everything on the right of the graph is more abundant with more adversity. Everything on the left is more abundant with less adversity. We ran a few different sets of models. So the first set of models was where we just had one focal adversity predictor in the model with a range of covariates. And then at the end, we also ran a model where we were pitting each of the adversities against each other, and all of the covariates were in there as well. So anytime you see all adversities in the model, that's from that model where all of the adversities were in the model. As is very common in this style of research, we used an exploratory Q threshold in order to look at our outcomes. And so what our Q threshold was 0.25, which means that we were willing to tolerate up to 25% of our results being due to chance or false positives. But what I'm going to show you here are the Q values that are associated with each of the results, and we can interpret these Q values. So this tells us the percent likelihood that this particular result is likely to be a false positive or not, and I have bolded those that we are the most confident in. And so looking just at the bolded ones here, we found that Streptococcus, which was higher in prenatal adversity, this is part of the normal microbiome, but several species are pathogenic, so maybe that's accounting for that association there. Ruminococcus, which was lower in prenatal adversity. Several species have been associated with immunity, both pro and anti-inflammatory functions, as well as with dopamine in the context of Parkinson's disease. Parabacteroides, which was low in postnatal adversity, is associated with health. So several species within Parabacteroides are potential probiotics, and it's lower in adversity, so that makes the most sense here. And then our result with the highest confidence was actually Clostridium sensu stricto, which was higher in the context of maternal childhood trauma. And this is a really interesting microbe because it's actually a butyrate producer. And again, we usually think of butyrate production as being a positive thing. But this particular microbe is a fairly inefficient butyrate producer that is usually outcompeted by 2 years of age. By more efficient butyrate producers. So maybe this is hinting at inefficient butyrate production in this group. So based on these data, again, it's very hard to get down to any functional significance here, but we're considering mechanisms of adversity impacts on the microbiome that involve immune function, that involve butyrate production, and we're testing those now using metagenomics data, which is going to get us down to a more detailed resolution. So thank Lord for us, the Gusto cohort actually found some money to get that metagenomics data. So we have that, or we're trying to figure out how to get 450 individuals' metagenomics data from one side of the world to the other, which is no small feat because they're huge, very heavy data. But we are trying to get that in hand now, so we should have some of that soon. So Friend also sees associations with some of these bacteria at a high taxonomic level with mental health of these young children. I'm not going to go into that for the sake of time today, but her paper is really wonderful and I encourage you to read it. So to summarise this section, maternal childhood adversity, maternal prenatal mental health and maternal postnatal mental health are each associated with the behaviour, brain and microbiome of second generation children. And for behaviour and brain, the intergenerational impact seems to be perpetuated through maternal, prenatal and postnatal mental health. So now that we know these intergenerational effects occur and that maternal mental health is so important to the outcomes, we've become very interested in the impact of childhood adversity on mum's transition to parenthood and particularly on mum's mental health transition through parenthood. So in the final part of this talk, don't worry, we're not going back to the start. I'm going to look at plasticity in pregnancy and how this might be a chance to address some of these intergenerational impacts of adversity. Okay, so here in this final part of the talk, I'm going to highlight the work of our second year graduate student, Paul Savoca, who's actually here today. You can wave, yay. That's Paul, he's great. And then our collaborators, Dr. Laura Glynn, who's at Chapman University, and your own Dr. Molly Fox, yay. So pregnancy is a critical period of development, which is also characterised by a heightened risk for depressive symptoms. So up to 12% of women will experience depression during their second or third trimester. And this depression risk is not evenly distributed in the population, so women who have experience with childhood trauma are at a greater risk for experiencing peripartum depression. Now, peripartum depression is important not just for its impacts on the mother and her own well-being, but it also has the potential to perpetuate cycles of adversity. So given that we're interested in these cycles of adversity occurring across generations, we're very interested in peripartum depression. Now, while there are some differences that exist between depression in the perinatal period and major depressive disorder at other stages of the lifespan, both are characterised by core negative affective symptomatology involving low or irritable mood, and both are characterised by abnormalities or altered functioning across numerous bodily systems, so the brain, immune, neuroendocrine systems. And several researchers, including most prominently Lisa Feldman-Barrett, have suggested that these widespread bodily impacts of depression really point to inefficient energy regulation at being at the core of depressive symptomatology, regardless of whether it's prenatal, postnatal, or at other stages of the lifespan. Now, energy regulation is supported by a process that we call interoception, which is really the way that the brain perceives, integrates, and models sensory information that is generated from within the body. And we know that interoception is also impaired in depression at other stages of the lifespan, so not specifically during pregnancy. So together with Paul, we wondered whether interoception might also underlie depression during the perinatal period. So when we think about pregnancy, one thing is for certain, and that is that interoception absolutely should change. There really isn't anything more certain than that. So this is a conceptual figure from a theoretical review that Paul is writing now, where we show that the metabolic demands, or we illustrate that the metabolic demands are drastically increased across the course of pregnancy and even still elevated in the postpartum period. And we theorise that under normal conditions, that is, not under conditions of early life adversity, that interoception should be able to become boosted during pregnancy to kind of keep up with these metabolic demands. Now, what we know from studies outside of pregnancy, so just in adult females, is that early life adversity is associated with a baseline kind of decrease in interoception. So if we just take a regular group of adult females and males, if they've been exposed to early life adversity, they'll generally have lower interoception. And so what we think might happen during pregnancy is that that difference between early life stress and not actually gets really enhanced during pregnancy. And this may be related to the increased peripartum depression risk we see in that early adversity group. So we tested this model in a study called Sensations of Motherhood, which was an online study started in February 2023 of 234 nulliparous women, means they haven't given birth before, aged 18 to 42 years. The pregnant group were in the second trimester of their first pregnancy that was carried past 8 weeks of gestation. So for all intents and purposes, their first full pregnancy. And the comparison group were not currently pregnant, had never been pregnant past 8 weeks gestation, and were not parenting any children. So the first thing we looked at were group differences between the pregnant and the not pregnant group in our primary variables of interest depression and interoception. So not surprisingly, we found that the pregnant group had higher levels of depression symptoms measured on the Edinburgh Postnatal Depression Scale. This has been shown time and time again. It's a high-risk period for depression. However, we also found that the pregnant group had higher levels of interoceptive sensibility. So this suggests to us that pregnancy might be some sort of sensitive period for this interoception learning, and certainly consistent with our hypotheses, we do see this boost in interoception during pregnancy. Across everyone in the sample, both comparison and pregnant group, we found that interoceptive sensibility was negatively associated with depression symptoms, which has been shown in previous studies. So depression generally is heightened when interoception is lower. Depression is decreased when interoception is higher. We found that childhood trauma moderated the link between pregnancy and interoception. So we analysed childhood trauma continuously, but here just breaking it down so you can see what the interaction looked like. So we found that there was a boost in interoception within the pregnant sample under conditions of low and moderate levels of childhood trauma exposure, but not under conditions of high childhood trauma exposure. And so then finally, we tested a moderated mediation model where we thought that interoception would act as a mediator of the link between pregnancy and depressive symptoms, but that this would only work under low and moderate levels of childhood trauma exposure. So the overall index of the moderated mediation was not significant but the conditional indirect effective group on depressive symptoms, so this mediation via interoception, was significant as expected for the lower moderate levels of childhood trauma, but not for the higher levels of childhood trauma exposure. And for reference here, the high childhood trauma exposure is really having two extremely traumatic events in your childhood. Okay, to summarise this final part of the talk, pregnancy might be a sensitive period for interoception under low adversity conditions might buffer against peripartum depression symptoms. And we think that pregnancy may amplify a pre-existing impairment in interoception within childhood trauma-exposed populations, leading to increased peripartum depression risk in that population. So this is a self-report study. We are trying to recruit individuals, if anyone knows anyone who's pregnant in their second trimester, for an in-person study where we look at not just self-reported interoception, but objective interoception measured through heartbeat detection tasks and whatnot. So exciting things on the horizon here as well. Okay, so here's the model that we started with. I've shown you today that caregiving adversity is associated with worsened mental health and physical health, and that this may be operating in part through adversity impacts on the brain-gut axis, including the microbiome. That these effects of adversity are also seen in the next generation, and we see these changes occurring at the level of the brain, physical health, mental health, and also the microbes of these children. And critically, that pregnancy may be a sensitive period of development for mind-body mechanisms, namely interoception, and that this is really important because interoception is linked with peripartum depression symptoms, which we think can help perpetuate cycles of adversity across generations. So this gives us a potentially important place to intervene to break these intergenerational cycles of adversity. So in terms of future directions, we are currently working on understanding how physical and mental health, particularly in terms of alexithymia, interact during childhood. We're really interested in how oral and gut microbiome communities interact with one another within an individual, especially to give rise to physical and mental health outcomes. And then finally, we're really interested in whether a mother's own microbiome during pregnancy relates to her experiences of childhood trauma, and how is that linked with her child's microbiome and also her interoception. So we plan a lot more work within the pregnancy period for the future. So I will end by saying an enormous thank you to the students in the lab. Jennifer Summers is the only one whose work I didn't really get to touch on today, which is a shame because she's a fantastic postdoc who's on the job market at the moment, and I can't say enough good things about her, to our collaborators and to our funders, and thank you all for paying attention.